My name is David Merrill. I'm a PhD candidate at University of Wisconsin. Today I'm presenting PathwayMultiomics, a method that I've been developing under the advisement of Tony Gitter. Our work is motivated by an application in precision oncology. It's based on the idea that pathways provide a useful vocabulary for describing cancer. We often describe a tumor in terms of the pathways that are abnormally active or inactive in it. This pathway perspective is clinically useful. If we know which pathways are abnormally active or inactive in the tumor, then we could, in principle, choose therapies that target those abnormalities. But before we can infer any pathway information about a tumor, we must first collect data from it. There are many kinds of data that we could collect from a tumor. For example, genomic data, transcriptomic data, proteomic data, so on and so forth. But it's increasingly common to collect multiple kinds of omic data and use them together. The idea is that multiple kinds of omic data can provide complementary views of a biological system, such as a tumor. It's like the old story of the blind people feeling the elephant. They may not do well individually, but in combination they could form a more complete picture. One example of a multiomic dataset comes from the Cancer Genome Atlas, or TCGA for short. It contains several kinds of omic data collected from more than 10,000 tumor samples. We want to infer pathway abnormalities using data like this. In order to do that, we frame the problem as a matrix factorization. Here's how that works. We take all of our multiomic data and put it in a big array where each row is a tumor sample and each column is a specific measurement. For example, one set of columns might be RNA-seq data, another set of columns might be copy number alteration, so on and so forth. But we assume that the data are generated by multiplying two much smaller matrices, X and Y. Each row of Y is called a linear factor, and the matrix X contains a low-dimensional embedding of the data. This is analogous to principal components analysis, where the rows of Y would be principal components, and X would be the transformed data. However, our matrix factorization differs from principal components analysis in important ways. We do the matrix factorization in such a way that each row of the matrix Y corresponds to a biological pathway. And if we do this correctly, then we can interpret the matrix X as pathway activity levels. Remember that our goal all along has been to infer which pathways are abnormally active or inactive. The matrix X will tell us that. So far, I've glided over many of the details that make this work. For example, we make sure that the rows of Y correspond to pathways by using a specially designed regularizer derived from known pathways. For now, suffice it to say that it's a combination of graph regularization and L1 regularization, and that we rely on the central dogma to model relationships between different kinds of omic data. I'm happy to discuss this in more detail with anybody who's interested. I've also glided over some of the extra layers in our model that allow it to accommodate multiple kinds of omic data. Different kinds of omic data look very different from each other. For example, RNA-seq counts may appear log-normally distributed, and methylation data may take values between 0 and 1, so on and so forth. Our model uses extra parameters and inverse link functions to account for these differences. It also has parameters to account for batch effects in the data. Again, I'm happy to discuss this in detail with anyone who's curious to know more. I'll point out that this model is comparable to several existing methods, but differs from them in notable ways. It occupies its own niche for now. It checks all of these boxes. This method is still a work in progress. But we have run it on the Cancer Genome Atlas, and the preliminary results seem promising. We've generated some interactive visualizations that you are free to explore at your leisure. Just follow this URL or scan the QR code. We've included a few kinds of visualizations. First, we've included scatter plots of the tumor samples embedded in our pathway basis. We also provide plots of the linear pathway factors, or in other words, the rows of the matrix Y, in our matrix factorization model. Lastly, we give a visualization that compares the pathway activity levels of different cancers and provides a sense for which ones are abnormal. Remember that that was the entire point of this whole exercise. As I've said, this method is still a work in progress. There are still improvements that we plan to make. Very importantly, we still need to subject Pathway Multiomics to a rigorous evaluation to determine whether it produces useful and reliable results. This includes a simulation study, as well as some evaluation on real data, from TCGA, for example. I'm eager to discuss this with you, or to hear any ideas you may have about model improvements or model evaluation. Lastly, I welcome you to look at our source code or check out some of the other work we've produced along the way. I'm also happy to chat about Julia with anybody who's interested. With that, I am finished, and I do look forward to talking with you at ISMB. Thank you for your time.